Welcome to the all viewers and well wishers of the world. The purpose of my lecture is learn the knowledge from the word, return the knowledge to the word. Motto of my life is learn, earn, and return. As I have learned a lot, earned a lot, now it's the time to return a lot to the word. The topic of today is antibiotics or antimicrobials classification. Classification of the antibiotics can be classified in several ways. So, the first, according to their action against infective organisms, spectrum of activity. According to the bacterial strain, they affect like streptococci, or Escherichia coli. On the basis of chemical structure as penicillins, cephalosporins, aminoglycosides, tetracyclines, macrolides or sulfonamides, among others, mainly classified by the mechanism of action of the antibiotics. Some antibiotics attack cell wall, some disrupt cell membrane, some attack at the ribosomes, and some attack on the other parts of the bacteria. So, some inhibit synthesis of the nucleic acid and proteins, polymers that make up the bacterial cell. Now, classification of the antimicrobial, we have 5 main groups of the antibacterial agents according to the site of action or the mechanism of action. Number 1 is inhibition of the bacterial cell wall synthesis like peptidoglycan or the component of the cell wall material is inhibited. Number 2, inhibition of the bacterial cytoplasmic membrane function, so disrupt the cell membrane which cannot hold the cytoplasmic contents of the bacteria. Number 3, inhibition of the bacterial protein synthesis and the ribosomal function, so the bacteria is unable to survive. Inhibition of the bacterial nucleic acid synthesis, DNA or the RNA, the brain of the bacteria, and inhibition of the bacterial folate synthesis, so bacteria cannot multiply and cannot survive. So, first of all, the inhibition of the bacterial cell wall synthesis. Mostly bacterial cell walls are unique. They have got the peptidoglycan that provides stability and protection for the bacterial cell wall and the shape of the bacteria. These antibiotics are the inhibitors of the cell wall synthesis. They act on the formation of the peptidoglycan layer. Bacteria that lack peptidoglycan like Mycoplasma, Chlamydia, they are resistant of these antimicrobial agents. These drugs are bactericidal as they weaken, lyse, and kill bacterial cell wall. These are called beta-lactam antibiotics as they all contain a beta-lactam ring, which is the main weapon of that antibiotic. By virtue of that, they do attack on the bacteria peptidoglycan layer and just lyse the cell wall and kill the bacteria. These antibiotics which inhibit cell wall synthesis of the bacteria, and here one can see the gram-positive bacteria, they have got a thick peptidoglycan layer, and here one is that, and in case of the gram-negative bacteria, there is a quite thin peptidoglycan layer as compared to this thick layer. Or in this diagram, one can clearly see and understand that thick peptidoglycan layer of the gram-positive bacteria and this thin peptidoglycan layer of gram-negative bacteria. And in my previous lecture and also lecture on anatomy, I have told you that peptidoglycan, these glycans are just like the bamboo-like structure, straight, and that support the bacterial cell wall. These bamboos, just like the ladder, are being connected by the peptide rings, and these peptide rings are again connected by a chain which is called protein binding protein, penicillin binding protein. Now, these antibiotic just attack on this chain and this chain which connects these two chains. So the ultimately result will be these will be lysed, and here one can see when all these standing rods between the two bamboos are lost, so the bamboos will be just weakened and drop and lies, and same is the case with the cell wall inhibitors. They attack the beta-lactam ring of these antibiotics that attack on this area of the peptidoglycan layer and weakens the peptidoglycan layer. Now, peptidoglycans are made of sugar backbone, as I have shown you the glycan, the bamboos, which is made of alternate molecules of the NAM and NAGs. If you just see the previous, this is NAG, the green one, and this is blue. These are alternate, as you can see. NAM and NAGs are alternate, and these cross chains are attached to the NAM molecules of the glycan rods. Every NAM molecule has a tetrapeptide side chains, peptido, I have shown you already. These are cross-linked by a pentapeptide chain. So here again you see the peptide chain, and that is being connected, the peptide chain, but the penta chains, these are being attacked by the beta-lactam rings. Beta-lactam antibiotics act on these cross links and side chains via beta-lactam ring, which weaken the cell wall. So this is the specificity of these antibiotics, that via their beta-lactam ring, they do attack at this point and weaken the cell wall. Now, continuing build-up of the material inside the cell put greater pressure on the cell membrane, and cell membrane now not supported by the peptidoglycan, which is a strong layer around the cell membrane, so it ruptures and cell contents leak out and the bacterium dies. Now, there are peptidoglycan synthesis. How this peptidoglycan is made, because there are certain bacteria which act not directly on the cell wall synthesis, but they act on the different components which make the cell wall. So coming to this point, here is the cytoplasm, and here one can see the red and the green and the dark green sugar molecules, and this peptidoglycan is made up of sugar molecules which combine with this amino acid molecules, as you can see. So sugar molecules and amino acid molecules that is being made here, it is just like the cement, the sand, the iron which makes a wall. So here in cytoplasm, all these things are made, manufactured, and these are transported via these anticaprinol. These are transporting all these things. As you can see, the sugar molecules attached with the amino acid molecules to the cell membrane, which is just enclosing or surrounding the cytoplasm. From the cell membrane, these are again transported to the cell wall, and here you can see the actual cell wall, which is being made up of the sugar molecules and the amino acid molecules, which are all manufactured here. So the compact cell wall is made, which is made of the bricks which have been made here, the iron, the sand, and the cementing material which make the cell wall. So the certain bacteria, certain antibiotics, they do attack on this side, and on this side to just ultimately make weak the cell wall. Now, the These drugs mainly include the beta-lactam antibiotics. I have told you all have beta-lactam ring in their structure. All bind to the protein penicillin binding protein PBS, as I have shown you that cross links of the glycans, these are being attached by a chain. So all these cross chains are being tagged by the beta-lactam rings. These receptors are situated at the cell wall and the resulting lysis of the cell wall. For convenience, we have divided into three groups: penicillin, cephalosporins, and the other groups which attack the cell wall. These are the carbapenem, monobactam, glycopeptide, beta-lactamase inhibitors, bacitracin, cycloserine, fosfomycin, and isoniazid. These are all basically attacking the cell wall of the bacteria. Now, one can see the core structure of the penicillin and the core structure of the cephalosporin represented by 1 and 2. Here, the red ring is the beta-lactam ring and the side chain which makes the new antibiotic and different names of the antibiotic. But mainly it is the beta-lactam ring which is red here, and similarly in case of the cephalosporin family, the main ring is the beta-lactam ring. Similarly, here one can see the carbapenem, they also have this beta-lactam ring and attached side chains, and similarly aztreonam that also have a beta-lactam ring and the side chain. So beta-lactam ring is the main weapon of penicillin, cephalosporin, carbapenems, and aztreonam as well. Now first, the penicillin. These are derived from the fungus Penicillium chrysogenum. Each member of the family of the penicillin shares a common basic structure, nucleus of 6-amino penicillinic acid, or it is conveniently called APA. 6-amino penicillinic acid, APA, which is the basic structure of every penicillin molecule, which contains a beta-lactam ring mainly with a side chain attached to it. Penicillin G and penicillin V are the unaltered products of the penicillium fermentation. Rest are having the attachment synthetically. Semi-synthetic penicillin are formed by addition of R group to the main 6-amino penicillinic acid APA ring, and most penicillins have little activity against gram-negative bacteria because they are unable to penetrate the outer membrane which is present in gram-negative bacteria and not in gram-positive bacteria. So these are mainly meant for gram-positive bacteria, and adverse reaction are 5% of the patient develop a hypersensitivity reaction, that is because of the penicilloic acid which is the product of penicillin. Now here it is the Penicillium chrysogenum which have produced this penicillin, which is having 6-amino penicillinic acid APA molecule, and here one can appreciate the beta-lactam ring which is the main weapon of this penicillin group. Now classification of the penicillin. For the clinical purpose, penicillins are divided into 5 main groups: narrow spectrum, moderate spectrum, broad spectrum, extended spectrum, and others. First, the narrow spectrum. It is further subdivided into narrow spectrum beta-lactamase sensitive, which do respond to the beta-lactamase enzyme of the bacteria, and bacteria capable of killing these antibiotics. Narrow spectrum beta-lactamase resistant or anti-staph penicillin. So the bacterial beta-lactamase is unable to do anything against these antibiotics. Narrow spectrum beta-lactamase resistant plus extended spectrum or anti-pseudomonal penicillin. These are very strong in the sense that they are capable of killing the pseudomonas bacteria also. Now, the narrow spectrum beta-lactam is sensitive. Their types is penicillin, benzyl penicillin or penicillin G, phenoxymethyl penicillin, penicillin V, procaine benzyl penicillin, and benzathine benzyl penicillin. And these are sensitive to the beta-lactamase ring, beta-lactamase enzyme if the bacteria do produce, and they are capable of just antagonizing these antibiotics. Now, the beta-lactamase resistant or anti-staph penicillin, the beta-lactamase enzyme produced by the bacteria cannot do anything against these. So these are very strong in the sense these are methicillin, oxacillin, cloxacillin, flucloxacillin, diclo, dicoxacillin, and nafcillin. Now, narrow spectrum beta-lactamase containing the beta-lactamase resistant plus extended spectrum or anti-pseudomonal penicillin. I have told you they are capable of killing the pseudomonas bacteria, and these include carbapenicillin, ticarcillin, temocillin, and carindacillin, capable of having such anti-pseudomonal activity. Now, modern spectrum penicillin or aminopenicillin, the second variety is narrow spectrum and moderate spectrum. These are the moderate spectrum. These contain ampicillin, amoxicillin, ricampicillin, and bifamcicillin. And the third variety is broad spectrum penicillin. These are co-amoxiclave, the combination of amoxicillin and clavulanic acid. Similarly, the combination of ampicillin and sulbactam by the name of unosine and other molecule in which the spectrum is very broad. So these are categorized under broad spectrum penicillin. Now, fourth variety is extended spectrum penicillin, or these are also called uridopenicillin. These are very strong penicillin, and these are piperacillin, azulocillin, mesalocillin, and the other variety is the mesalinam, mesalinam, or salbenicillin. These are the other varieties which does not fall in all these four varieties, so that have been just made one extra and separate. It is said to be that it is better to be hated for what you are, but you are original, whatever you are. You are not posing anything. Than to be loved for what you are not, what you are posing, what you are just showing yourself, but actually from inside you are not like that. So it is better to be hated. The classification. The second group is cephalosporins. These are isolated in 1948 from the fungus Cephalosporium, beta-lact structure like that of the penicillin, but here in place of the 6-amino penicillinic acid APA, they have a nucleus of 7-amino cephalosporanic acid instead of APA, chemically modified to produce a family of various related antibiotics given to the patient with penicillin allergies and the penicillin resistant cases, so they do respond well to the cephalosporin. They are grouped as first, second, third, fourth, and fifth generation according to their spectrum. They are more active against gram-negative bacteria than gram-positive bacteria. Some are very good against gram-positive bacteria, like first and second generation, but the rest of the generation are more against the gram-negative bacteria. Here one can see the cephalosporium, the fungus which have made this cephalosporin possible, and again here one can see the beta-lactam ring, the main weapon of these antibiotics, and Now, the semi-synthetic cephalosporins are prepared by attaching the different chemical groups at R1 level and R2 level. Here one can see the beta-lactam ring and the side chains are attached at R1 and R2 to make the different cephalosporin, but the basic structure is still the same, 7 amino cephalosporanic acid, and that is the site of cleavage by the bacteria beta lactamase or by the acid, which are bacteria capable of destroying that beta ring and making that antibiotic ineffective. Now, first generation cephalosporin, these are more effective against gram-positive bacteria than gram-negative bacteria, and these include cephalexin, cephradine, cephalothin, cefadroxil, cefazolin, and the second generation cephalosporin. They act against many gram-negative and many gram-positive bacteria, so they have a bit balanced spectrum against gram-positive and gram-negative bacteria. These are cefaclor, cefuroxim, cefamendol, and cefoxetine. The third generation cephalosporin, these are effective against gram-negative bacteria more and reach to the central nervous system, so capable of treating the CNS infection. These are ceftriaxone, cefotaxime, cefpodoxime, cefixime, cefoperazone, and ceftazidime. And fourth generation cephalosporin are more effective against gram-negative pathogens and less against gram-positive. These are cefepime, cefpirone, cefazopran, and cefpinone. The fifth generation cephalosporin, that is more effective against gram-negative pathogens, and that contains one molecule, that is ceftobiprole. Now, other groups which are capable of inhibiting the cell wall, or we can call it cell wall inhibitors, they are carbapenem, including imipenem, meropenem, monobactam like aztreonam, glycopeptides, including vancomycin and teicoplanin, beta-lactamase inhibitors like clavulanic acid and sulbactam, bacitracin, cycloserine, phosphomycin, and isoniazid. So the first, carbapenem, these are the beta-lactam antibiotic, biological sources, streptomyces, cattleya, as one can see, effective against a wide range of gram-positive and gram-negative bacteria. They are resistant to the hydrolysis by the beta-lactamase enzyme produced by the bacteria, and these include imipenem, meropenem, ertapenem, and ferropenem. Here one can see the molecule, and they all contain beta-lactam ring. By virtue of that, they are capable of destroying the cell wall of bacteria. The next is monobactam, and it contains aztreonam, the main antibiotic, beta-lactam antibiotic. Biological sources, Chromobacter violacum. There is no fused ring attached to beta-lactam nucleus, single beta-lactam ring, as one can see, and spectrum only for the gram-negative and aerobic bacteria like Enterobacteriaceae and Pseudomonas, so very effective against these tough gram-negative bacteria. Number 3 is glycopeptides. These are biological source is Streptomyces orientales, as you can see the fungus, to treat serious infection such as endocarditis and septicemia caused by the gram-positive cocci. They are multi-resistant to the other drugs, and that is group contain the vancomycin, teicoplanin, and talavancin like molecules, which are very tough molecule against the bacteria. Number 4 is beta-lactamase inhibitors. These agents have negligible antimicrobial activity, although contain beta-lactam ring, but they are not having very strong beta-lactam ring, which is capable of killing the bacteria. But these bind with the enzymes beta-lactamases produced by the bacteria, so prevent inactivation of the beta-lactam antibiotics. So they just combat with the beta-lactamase enzyme produced by the bacteria so that they can kill the bacteria by the beta-lactam ring. They have high affinity for and stability to the bacterial beta-lactamases. They are co-administered with beta-lactam antibiotic. They are grouped into three groups: clavum, panum, and celestatin. Here one can see the clavum. They include clavulanic acid. Biological source is Streptomyces clavuligerus. Spectrum is effective against gram-positive and gram-negative bacteria. Mode of action is they are suicide inhibitor of the beta-lactamase enzyme. Number B is the panum. They include tazobactam and sulbactam. And the third is celestatin. These all three are used in combination with other beta-lactam inhibitors, but their main function is that they inhibit the beta-lactamase enzyme produced by the bacteria for their survival. Now, beta-lactamase inhibitors combinations are co-amoxiclav, which is a combination of amoxicillin with clavulanic acid. Ticarcillin is just combined with clavulinate. Similarly, ampicillin is combined with sulbactam, and piperacillin is combined with tazobactam, and imipenem is combined with celestatin to enhance their antibacterial activity. Now, the fifth antibiotic capable of killing the cell wall or the lysis of the cell wall is bacitracin. Biological source is Bacillus subtilis. It is effective against gram-positive and gram-negative bacteria, but mainly the gram-positive bacteria, penicillin-resistant staphylococci, are sensitive to them. They are too toxic for the systemic use, but used in the topical preparation. Here one can see the bacteria, Bacillus subtilis, and here it is the formation of the different products by these bacteria, Bacillus subtilis, and it is mostly used as bacitracin ointment. Topically, it is very effective. Number 6 is the cycloserine, an analog of D-alanine. It interferes with the enzymatic conversion of L-alanine to D-alanine in cytoplasm. So, synthesis of the peptidoglycan cannot occur. I have shown you in the previous slide that the different subunits of the cell wall are made at the different level. So this antibiotic just do have the effect at the manufacturing of the cell wall during its cell wall synthesis. So that makes the cell wall ineffective for having the protection against the antibiotic. It is used as a second-line drug in TB with multi-resistant strains of Mycobacterium TB and also in the recurrent UTI infection. Number 7 is the fosfomycin. It is a broad-spectrum antibiotic, blocks formation of the peptidoglycan. It is effective against gram-negative bacilli, mainly used for treatment of urinary tract infection, especially soluble trometamol formulation, phosphonic acid derivative, phosmidomycin is available, and it is effective against gram-negative bacteria. Now, isoniazid, also known as isonicotinic acid hydrate, it is capable of stopping or blocking the synthesis of the mycolic acid in the cell wall of the mycobacterium, tuberculosis bacteria, an antibiotic to treat active tuberculosis, a bactericidal agent active against organism of the genus Mycobacterium, like Mycobacterium tuberculosis, Mycobacterium bovis, and Mycobacterium kansasi. These are effective against the mycolic acid, the cell wall of the mycobacterium that is being killed by this antibiotic and causes the lysis of the cell wall of these kind of bacteria. Now, there are different levels of the cell wall inhibition, and in the synthesis of the cell wall, I have told you also the different level. So, the drugs that inhibit biosynthetic enzymes in making of the cell wall, these are phosphomycin and the cycloserine. The drugs that combine with carrier molecule, so uh, during its manufacture, it cannot be carried to the cell wall proper from the cytoplasm, that is the bacitracin. Now, the drugs that combine with cell wall substrate, that is just in place of the iron, in place of the bricks. So, these are going to just join in place of the bricks. So, the defective cell wall is formed that is very easily lysed. So, these are the antibiotics which do that, that is the vancomycin. Now, a smile is the best makeup a girl could wear, that is said by the Marilyn Monroe. Whatever the makeup you have done, but a smile of the woman is the best thing which a girl can give you and make you just astonished.